year 2011, scientists at Sorbonne University Pierre Marie Curie and the University of California, Berkeley discovered a way to edit the DNA of any living organism. They called this new discovery CRISPR. Their breakthrough came when they found that they could reprogram a bacterial immune system to search out and cut any DNA sequence of their choosing. This opened the door to previously unthinkable types of genetic engineering. Simply program the CRISPR guide RNA with the exact sequence of letters you wish to cut, and then the Cas9 protein will cut the target DNA in that exact spot, like a pair of molecular scissors. Shortly thereafter, the US military unleashed their first CRISPR project, codenamed Big Red, a genetically modified giant red dog capable of sniffing out rare earth minerals.
Stop. At some point there, you switched from reality to that movie Clifford the Big Red Dog. But you are actually right. CRISPR is a really easy way to edit DNA. But as we've been talking about in all of these episodes, that is a useless skill unless we understand the complex language of DNA. Like, yes, technically we could use CRISPR to edit the DNA of a puppy, but we don't know what to write in order to make it the size of an ice cream truck. So before we start designing entirely new monstrosities, first we need to decipher the DNA code. And luckily, CRISPR is really useful for that too. Alright, so what are we doing in here?
We're going to inject some zebrafish embryos.
Inject them with what?
CRISPR.
So this is the actual CRISPRing.
That's right. This is where the sausage is made. This is the fish facility. In here we're able to house many thousands of zebrafish for genetic experimentation. You can see fish with long fins here. Have mutations in potassium channels that cause their fins to get really long. So these fish have shorter fins from the same screen. These are also zebrafish, believe it or not, but they have, I believe, 3 different mutations that gets rid of all their pigment.
You can actually already see some of the eggs on this side, like white little orbs right there. Those are the developing fish embryos, and that's what we'll be injecting.
To time the matings, what we do is we set up the fish with a barrier between them, a male on one side and a female on the other. When it comes time to introduce them, you can remove this plastic barrier and they'll come together and hopefully produce embryos. In the wild, zebrafish spawn along the shores of their freshwater environments, and so we add this mating grass to kind of try and simulate as best we can their natural spawning environment.
You seem like you're phoning it in a little bit with the ambiance.
Zebrafish embryos develop fairly rapidly, so within about 48 hours or so, they'll hatch as a free-swimming larva. So we got to make sure we catch that single cell within a few-hour time frame so that we can inject the CRISPR.
So I'm about to pipette our CRISPR into the injection needles. So this is our CRISPR that I'm going to load into our injection needle.
Why is it red?
Better see where we're targeting, and also just for camera sake. It's not actually red.
Well, so CRISPR has made it really easy to knock out individual genes and then see what happens.
Genes like zebrafish 1111, a sequence of 5,955 DNA letters found in every single zebrafish. And it's not just zebrafish. Very similar 1111 genes are actually found all over the animal kingdom. Fish, cats, giraffes, people, and lots of other species all have a 1111 gene. That can't be a coincidence. If pretty much the same piece of code is showing up in all these different species, it must do something important. We just have to figure out what that thing is. Our first clue comes from the Antarctic Ocean, where you can find fish with 1111 and fish without 1111. The fish with 1111 have normal, hard skulls, while the fish without 1111 have much softer skulls. So this is our hypothesis. The TRPL11 gene has something to do with skull density. 15 years ago, that's where this story would have ended. You know, a nice hypothesis. But now we have CRISPR. We can use CRISPR to delete the TRPL11 gene in our zebrafish and then, just see what happens. If the skulls get soft, we were right. If the skulls don't get soft, we were wrong.
How big of a deal is it to actually be able to test out some of these things?
It's a huge deal. I mean, that's one of the, a lot of times people, when they talk about CRISPR, they focus on the editing aspect in terms of engineering organisms to do something that they don't already do. But equally important, in my opinion, aspect to this, which is just it has made genome engineering so much easier that everyday biologists studying any number of traits can now go in and start testing their hypotheses with much more rigor and ease.
What I usually do is I look through the microscope and I just try to line them up as best I can.
Okay, that's what CRISPR looks like, I guess. How does that fluid actually work?
You obviously weren't paying attention earlier. I explained all of this in the beginning of the video. Plus, all of this information is easily accessible on my website, drpyramidesquire.geocities.com.
I don't want to go to your website. Can you just explain it again?
Fine. In the year 2011, scientists at...
No, not that part. Just the part about how CRISPR actually works.
The CRISPR fluid is actually made up of two different chemical molecules, the CRISPR guide RNA and the Cas9 protein. The Cas9 protein is the pair of scissors that cut the target DNA, and the guide RNA is the roadmap that tells the scissors where to go. All an individual scientist needs to do is design a guide RNA with the specific set of letters they wish to cut, order that guide RNA off the internet, and then inject their CRISPR formulation into their test subjects.
Interesting. I wonder what the guide RNA looked like for deleting our TRPL11 gene.
Here we have zebrafish TRPL11, the full coding sequence, starting here with ATG, and then a whole bunch of ATs, Cs, and Gs all the way down to the end down here. If we're interested in knocking out TRPL11 function, the best place to start would be near the start of the protein, because if you damage it early on, chances are it won't go on to make the rest of the protein. So we have start of the gene going on to the full length. If we want to knock it out with CRISPR-Cas9, we can introduce a guide RNA that has the exact sequence matching the region that we want to introduce a deletion. This is the part that makes CRISPR so valuable, is that you can design a set of only a few base pairs here that match the region of genome you want to target for deletion and direct Cas9 right to that place in the genome. When Cas9 is directed to this location in the genome, it will introduce double-strand breaks, which will be repaired by the host genome. And in doing so, there will sometimes be errors that occur, some of which introduce deletions like you see here. And it results in the protein being terminated very soon thereafter.
So this is a complete turning off of the TRPL11 gene.
Right. We've more or less removed it entirely.
Now it's the moment of truth. It's time to find out if removing TRPL11 actually made our fish heads softer.
What we find is that loss of TRPL11 results in a huge decrease in overall skeletal density. This normal-looking fish skull looks a little bit like this. We're actually looking down at it from on top. So we're looking at sort of the skull here. And without TRPL11, there's hardly any dense tissue at all, actually. So we now know that TRPL11 really is important in regulating skeletal density in fishes. This gene is also found in mammals and in humans, and what we now know is that this is also really important in regulating human skeletal density as well. And so, again, by studying some of these different fish species, like Antarctic fishes as well as zebrafish, we can understand how genes are regulating some of these traits that are also important in understanding human disease as well.
Next time, in our series finale, we're going to take everything we've learned so far and use it to cure sickle cell disease with CRISPR.